So today's topic we are going to deal with is DNA replication. So what do you mean by the process of DNA replication? Okay, so let's start it with an activity. Here you can see the image of a spring, is it? This is an image of a spring. So now what I'm going to do is, see now what difference you can see. Yeah, you can see the multiple copies of the same spring over here. That means this spring has been copied to two different springs, is it? Yes. Now, can you relate the structure to any material in our cell? The structure of the spring to any material in our cell? Yes, it's DNA. We have DNA that has a similar shape to this spring. Okay, so this is DNA. You can see it has a spiral structure, whereas the spring is also having a spiral structure. And now, do you believe that when the cell divides, DNA is also copied to daughter cells? That means as the spring divided into two cells, do you believe that in our cell, this DNA is also being divided into two daughter cells or any number of cells? Do you believe that it is copied? Yes, let us check onto it. Okay, so if DNA is copied into two cells, yeah, it will be like the same copy of the parent cell. This daughter DNA cells will also look similar to the parent DNA cell. Okay, so this is the parent DNA cell and these two are the daughters. Okay, so this process is known as DNA replication. So today in our topic DNA replication, we are going to learn how the process of DNA replication taking place and also what is the role of the enzyme DNA polymerase. It is an important enzyme that helps in DNA replication, DNA polymerase. Okay, now DNA replication. It is the process by which a double-stranded DNA molecule is copied to produce two identical DNA molecules. Yes, that we have seen in the slide. The identical copying of a parent DNA molecule. Okay, a parent DNA molecule is copied into two identical copies. Okay, check that word. Two identical copies. Okay, identical DNA molecules. That is known as the process of DNA replication. And this takes place through 4 steps. So, it is an important point. The 4 steps through which DNA replication takes place: DNA unwinding and replication fork formation. Then a primer will come and bind onto the unwinded strand. Then elongation will happen and then termination will happen. So we can deal it with one by one. The first point, unwinding of DNA and replication fork formation. So you know that this double-stranded spiral structure is the DNA molecule, and before DNA replication, this structure will unwind like this. Okay, this spherical structure will unwind like this. Okay, so that is known as unwinding of DNA, and replication starts at a particular sequence. Okay, replication always start at a particular sequence. This is that sequence, and since it is the start, it is known as origin of replication, and the double-stranded DNA unwinds with the help of helicase enzyme at this point. Okay, so this blue color structure, this is known as helicase enzyme. Helicase enzyme helps in unwinding this strand, and now the helicase enzyme will come and bind onto this origin of replication, and then what happens? Here the unwinding starts and the unwinding of the two strands take place. Now the unwinded DNA now looks like a fork. See what is the shape you can see here? The unwinded DNA. This is in the shape of a fork, is it? Yes, that is known as replication fork formation. So before the replication starts, all this process take place. What happens? First, the helicase enzyme will come and bind onto this origin of replication, and then what happens? The helicase enzyme will act and the unwinding of the DNA takes place, and then due to the unwinding, a replication fork, a fork structure is formed here. So that is known as a replication fork. Okay, so this is the first step before DNA replication. Now you can see the second step, that is primer binding. Yes, the duplication starts by the binding of an RNA primer. So primer by the word, it means it is a starter. Okay, so it helps in the starting of the particular process, that is replication. Okay, so RNA primer, it is an RNA coding sequence. The RNA primer will come and bind onto the DNA unwinded strand, and from here the duplication starts. So wherever the primer comes and binds, from there the duplication starts. So this is a primer sequence. You can see here, this is a primer sequence that will come and bind onto the 3' end of the DNA strand. So these blue color strands, no, these are the two DNA strands you can see, and see, the strands are complementary to each other. 3' to 5', here 5' to 3'. Okay, they are anti-parallel. So that is why it is marked as 3' to 5', and on the second strand it is starting from 5' to 3'. So it is anti-parallel. But you can see that the replication always starts from only the 3' end. Okay, only the 3' end. So you can see here that the 3' at the 3' end, the primer sequence, RNA primer sequence is come and binding. Okay, then the primase enzyme synthesize RNA primer. So who synthesized this RNA primer? The enzyme primase, and the primer always binds at the 3' end. So that is an important point. The primer will always bind at the 3' end. Okay, it will always bind at the 3' end. So after the binding of the primer, the replication starts from the 3' end. So the next step is elongation. So after the binding of the primer, what should happen? The elongation of the strand will happen now. Okay, here in this figure you can see that from the 3' strand, okay, after the primer binds, replication will start and it follow towards the 5' end. Okay, so here also you can see it starts from the 3' end. That means this end, and it will go on to the 5' end. But here you can see that two newly strands are formed, and the one newly formed strand is continuous. That means there is no breaks in between, is it? But here you can see in the second strand there are some breaks, is it? Yeah, let us look onto it. So here, what is the first point you can see in elongation? An enzyme DNA polymerase 3 carries nucleotide and binds at 3' end. Okay, so after the binding of the DNA primer, now the process of replication or the elongation of the rest of the strand is going to start, and during that elongation, the DNA polymerase enzyme, that is DNA polymerase 3 enzyme, helps in elongation. So now the DNA primer will go, and the DNA polymerase 3 enzyme will come and bind at that point, and it will bring the nucleotides to the strand, and the strand is now continually synthesized. The DNA polymerase elongates, thus forming DNA polymerase elongates and forms a new leading strand from 3' to 5' end. From 3' to 5' end, a leading strand. This continuous unbreaked strand is known as leading strand. Okay. Now, the new leading strand is continuous. Yes, the new leading strand is continuous. Okay. And next, what happens? In contrast, see, you can see here on the other side, an uncontinuous strand is formed. Let us look on to how it is formed. The primer binds discontinuously along the lagging strand. Okay, so lagging strand forms away from the replication fork. Here you can see the leading strand is forming towards the replication fork, and the lagging strand is forming away from the replication fork. So I will show you in another color. Okay, so here you can see the leading strand is forming towards the replication fork, whereas the lagging strand starts or formed away from the replication fork. Okay, so this is the difference, and the leading strand is always continuous and the lagging strand is always discontinuous since it is forming away from the replication fork. And when this lagging strand is forming, DNA polymerase 3 helps in elongation. The same enzyme is helping. And since these fragments are, see, breaked over here, this is known as Okazaki fragments. Okay, what is the name? It is known as Okazaki fragments. Okay, now here you can see this is the parental strand, and these gray color strands are the parental strands. So the replication starts from 3' end. So the leading strand stands from it is towards the replication fork. See, the arrow is showing that, and whereas the lagging strand formation is from it is away from the replication fork. That means from the 3' end to 5'. So always remember the replication starts from 3' end to 5' end, and the two strands of the DNA are always anti-parallel or complementary to each other. Okay, and when you draw these figures, first you mark the 3' and 5' end of the two strands, 5' and 3' end of the two strands, so that the rest of the portion will be easier for you. Now, let us look into what happened to these Okazaki fragments. Now, these Okazaki fragments should join over here, right? So, for that, one enzyme will come and help in the joining of these Okazaki fragments. That enzyme is known as DNA ligase. See, the enzyme DNA ligase joins the fragments, Okazaki fragments. So now this lagging strand also will become continuous. Okay, so now the fourth step is termination. That means this elongation or this formation of the strand, a new strand should stop now, is it? So what happened? Here, look at this image. After the formation of the strand, what happens? The old strands will curve and come and join at a point. Okay, at this point it will divide. Okay, at this point it will divide. Okay, so after replication, now the fork meets at a point on the same stretch of DNA. Okay, so I will show you once again. Here, after the formation of the replication fork or the new DNA strand, what happens? It will meet. The two strands will come and meet at a point, that is this point. Okay, the new strands will come and meet at this point. This is known as termination, and it will break over here and it will separate. So this is known as termination. So what it is known as? Termination. So this is the total process of DNA replication. A DNA strand will unwind and form two strands. Okay, and then what happens from the 3' to 5' end? Here, this is the leading strand and this is the lagging strand, and in the leading strand the strand will form from 3' to 5' end as continuous, and on the lagging strand what happens, it will form away from the replication fork as Okazaki fragments. Okay, so this process is known as DNA replication, the copying of DNA, parent DNA into two identical daughters. Okay, or the two identical copying of the parental DNA is known as DNA replication. Okay, replication. I think now the topic is clear for you. Okay, thank you.